Have to be closer. Okay. So today is essentially about getting to 3D structures. Let me repeat. Whenever you see a model, that's sort of the lesson from comparative modeling, but this is sort of true for any kind of prediction method. When you see a model, ask yourself what in that model is most likely right, and where are the mistakes. Both of those questions typically help you to interpret your model, and that should be a question you ask before you ask what is it that I learn in terms of protein function, for instance. So first begin with the simple things. Structure genomics, structure determination is the topic today, structure by experiment. So quick run through, a slightly outdated slide, but in terms of percentage that remains the same. PDB currently has 100,000 records. This was done at a point it was 84,000 records, of which 70 of these 84 were 89%, roughly 90% were in fact of these here are by X-ray crystallography. This 1% is 1% of all the known proteins. That's what that number meant. NMR 10%, electron microscopy 1%, and that number here is increasing slightly. Let's get back to crystallography. So the idea is you take your protein, you grow crystal. How do you do that? How do you get a protein into a crystal? So you need a very high concentration for a crystal to form. So a crystal, essentially the definition of a crystal is you have something that has a repeated, sort of has a unit and repeats this unit all over again, right? That's the way you form a crystal. You know it from the diamond contact in school, right? The diamond shape comes from the, in fact, if you, there's a nice museum here in Munich. The Museum from Mensch und Natur is what it's called, in the castle. What is this castle called? That's the one, right. So in the Nymphenburg Castle, you have this museum, and in this museum you really see how you can, on a microscopic level of seeing the stones, see the difference of the molecular structure in the difference of the stone shape. So they have nice images where you see the molecular shape, and then you see the real stone, and you see a cut through the stone, and that you see really how it breaks and how the molecular shape is visible on how the stone forms. That's amazing in a crystal. But it's the same thing here. So you need something that's repeated over and over again, and for that you need a high concentration of protein. Do they exist in high concentrations in the cell?
Depends on the protein.
That is correct. So in fact, there are some proteins that were scooped out. Early crystals were partially done in some cases by things that were essentially scooped out of the protein, of the cell, because they get them in extremely high concentrations. But that is the exception. Most of the cases, what you have to do is you have to take, you clone it, you purify it, you make it aggregate, express, and you get a lot of this. And this is the major challenge here for crystallography. Get a high amount of clean, clear protein and make it form a crystal. So now we get into something else. A lot of crystallography, in fact, has to do with something that is more a cooking book, art. Growing crystals, all of these large labs, the Perutz lab, that was those were the first 2 year Kendrick Perutz. I don't know from who this structure here is. That really is bad. Should have the structure listed somewhere. But today's big labs, they all have their expert in crystallography, in crystal growing. So those are people who have an extremely good hand at growing crystals. And the way they do it is they simply try a lot of conditions. So somebody gives them this highly purified proteins, and then they put some solvents in there. Solvents or additives, let's call them additives. Typically, they contain metals, all types of metals, and all kinds of other substances. Typically, what you achieve with those is you make the protein bind and aggregate in repeated single cell units. Then once you have these crystals grow, and every crystallographer typically when they talk about their protein structure begins with a crystal. So if you were a crystallographer and you would see this crystal, you would see in that crystal something about the quality of the structure. So I don't. For me, when I see these talks, for me they typically look all the same. Although I've now seen many of these talks, but really the crystallographers, they immediately get a feel. So Wayne Hendrickson was my colleague was sitting next to me, next lab to me in Colombia, and he's just one of the person who looks at a crystal and immediately can tell you the resolution. Maybe sometimes he's wrong, but you really get an intuition, and all you need is this figure essentially. Okay, then what you do is you shine X-ray beams into it. The X-ray beams scatter, and that again, when you go to the subway here, not to the bike, in the subway there's the diffraction, the Nobel Prize that was won for X-ray diffraction. Essentially, this experiment is in the subway shown, and that is the same way it does. So essentially, you scatter the light that produces an electron density. From the scattering, you in fact infer then the 3D structure. I'm not explaining how X-ray crystallography works. I'm just giving an idea, okay? It's very complicated to get from that electron density. So to retrace where the original crystal is, the principle is written there. The principle is the Bragg reflection. That is a simple principle. But the way to really get it out is highly non-trivial. And there's a lot of math, in fact, underneath this mapping. Now, from that electron density map, that's the raw data if you want, you get into the atomic model through fitting. Here is an atomic model, 4 different levels of resolution. This is 3 angstrom, and this resolution, in fact, means how accurate is it. And what it means essentially is an RMSD. It's a root mean square deviation that sort of is assumed to be the mistake you make. So here's 3 angstrom mistake, 2.72 and 1. So this is the highest resolution, and you see that while here blue is the electron density map, and you see that the ring in this, here you have a mesh of blue, and you do see that the ring fits into this net, right? And that this all is a good fit, but you could also move it slightly around, and you would still somehow have a fit. While this one here, the 1 angstrom resolution, you see the ring. You really see the blue tell you exactly where the ring sits. Okay, that's the difference between 3 and 1. This level, so I said we have at this point about 100,000. I don't know how many we have of these, but certainly this is in the ballpark of 100, typically less than 100. A few years ago, so there was just 1 or 2. This is very new that you can reach that resolution, and it's really complicated to achieve. This is what today is considered not good enough for X-ray crystallography, and this is the level where most structures sit, in between. Okay? Now we get to NMR. The big issue in the crystal structure is that you have to grow a crystal. NMR, you simply take the protein in the solvent. In solution, it's a native environment in some sense. It's not the native environment as in you put an entire cell here into the NMR tube. But all you need to do is essentially get your protein, put it into solvent. Okay, you need still to get your protein, you still need to get to express a significant amount, but no longer that much protein that you can grow a crystal. You no longer need to grow a crystal. You no longer need to do something that may not be like its native environment. You keep it in solvent, and proteins live in solvent. Now what you do, and again, what we're talking about is 10% of today's structure. Now what you do is you put this NMR tube into a magnetic field. In the magnetic field, you start a big magnet. The big magnet excites all the protons, and then they fall back onto their original state, the demagnetization. And that difference is what you measure. And from that difference, you measure actually couplings between protons. That you can translate to couplings between residues. Now we get into a little bit of the problem that you measure a coupling between leucine and an isoleucine, but you don't know which leucine and which isoleucine. So you need additional experiments in order to solve that problem. So you go one by one. You begin to, in fact, see that is a leucine-isoleucine contact. Where in the protein could it be? Let's do another measurement, and then I figure that one out. So X-ray crystallography is the type of experiment where you grow the crystal, then you get to a large beam. Large beam, they are typically really large. In Germany there's one, in Hamburg, at the DESY. Most of the experiments now are done in CERN, in Geneva. So in this big, so most of these large high energy accelerators in physics are today used, in fact, a lot of the money that they get comes from biology. And that is X-ray crystallography. Because the reason why they have these large beams is because they are very focused. Extremely unique. So anyway, the point is, you take your crystal, you go to that big beam where you rent your time, and then from there on to the structures maybe a week. Here, from having the first set of experimental data to the first assignment may take an expert a month. Then there comes the second. Then comes, so at some point, a lot of the NMR structures, by the way, have been done around a group of Guy Mantuglione, and I'll get back to that in a minute. And Guy keeps saying that today we're so fast that an expert can do it in some cases in 2 months. Okay, that's sort of the difference. The difference is here, you spend 2 months and the other one a week. And the other one is a postdoc. Here it's a professor or somebody who's really deep in the field. The disadvantages, you don't need here the advantages, you don't need a crystal. Okay? The magnets that you need are big. So when you go to a hospital, that is in fact the same principle. You do exactly the same thing, except for the magnets are weak. And there are these, I don't know whether you have ever seen them, ever been in one of these tubes. They feel big and they don't, it doesn't feel good to be in them. But these things are really, really huge. So this is the person, and that's a 900 megahertz machine. So today they have 1100 or 1200 machines. They are way bigger than this. That, if you put, so first of all, this thing you cannot put you cannot put everywhere, okay? So I remember that there was an issue where to put that. Manhattan is built on concrete. That's why you have these high sky risers. And it's like sheet, rock sheet. So it's really, really, really tough. And even there, it was not that easy to find the right spot for it, okay? That is number one, just static. These things are heavy. Number two, you need to have a huge environment. And you see this one here. All of this is a Faraday cage. In fact, that is 4 stories high or 5 stories high. Now the machines are bigger, as I said. This is sort of the New York City College, New York Structure Biology Center at its beginning here. And so there's a lot of shielding going on. And then in this room, you have anywhere in this room, you have an absolutely safe future for your credit cards. You will have no more problems with that credit card. We'll just not have to try it. Anything that comes in here that is magnetic material will change. So your passport, so these kind of things typically happen that somebody carries a passport and they will not be able to easily leave the country afterwards. So that has happened here in this particular place. To me it happened only with a credit card. Anyway, and then actually that was before, the machines just came and they had not really been started, and I didn't take it seriously. I mean, nobody has really run them. So anyway, these things are really big and impressive. What you get out is you get a lot of coupling constraints. These coupling constraints give you an idea about the 3D structure, but not everywhere, not always. So what you get in the PDB is something like this, where in fact you do not have one solution. You have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and so forth. You can count it, right? These are all different solutions. And they are different solutions because what you do is you begin from the constraints, the couplings, the pairs, the interactions between residues, and you ask, given these constraints, what's the most probable model that I get? And that is a Monte Carlo simulation, essentially. And in this Monte Carlo simulation, you have different starting points and you have different endpoints. So somehow you can be trapped in local minima. So you have different solutions, and these are the 13 or whatever I just counted, 13 different solutions that are compatible with the experimental data you have. NMR spectroscopists say, when they see something like that, okay, wherever the protein is not moving, here in this core, where you should see the strands. Essentially what you have is a Greek key kind of a motif with a helix here. NMR spectroscopists would argue that is where the protein is stable and has to be the same shape. While here, at the ends, what you see is a dynamic change. My model is not well defined because this thing moves in solvent. And moving in solvent actually reflects its dynamic feature as a native protein, because it has to move. So they would argue that the differences that they see pertain to functional variation or functional movement. Okay? But this in fact tells you a lot about function. Extra crystallographers would typically argue, guys, what you have are a bunch of mistakes here. You can't decide on one solution, and because your data is not good enough, you sell that as a study of function. In our crystal, we have enough information about function. Where do you fall on that debate? Who's right? Since that is a movie only for you guys, we can easily discuss this. I'm not comfortable going even that far. My typical internationally.
This is an interesting point of view. It gives some good insight into this structure. I think more than the X-ray crystal.
So the problem is, my background really is in physics, and sort of the more modern physicists are the NMR spectroscopists. It's just a cool way of doing it. And these are clever guys, and they typically sell their argument with a lot of clever arguments. It's also true, we know that historically when NMR started, it was so complicated that people made many mistakes. And the first NMR models that were deposited in the PDB have all been removed because they were all wrong. So that does not help the community a lot, right? So somehow, but let's get back to the question. In a cell, one part of the argument is in the cell, proteins are like they have them here in solution. They don't have the density of a crystal, right? Is that true?
Yes.
Maria, Sebastian, what do you say?
Depends on solvents, if it's similar to...
Just water.
Yeah.
Yes. In fact, again, they try to make it native-like as possible. Yes. And as I said, it's pretty much water. Is that true? What's the density in a cell? That's not quite true, actually. So the density in a cell is remarkably high. For most of my career, I have imagined a cell, most of the time that I worked on structure prediction, really imagined a cell to be a bag of water. And in that bag of water, you have things swimming around. But the image of a clockwork, the image of something that has a solid state, is closer to the truth. It is almost the density of a solid state. Yes, there's water in there. Yes, every protein sits in there somehow folding in water. But the environment is extremely dense. In fact, the idea of swimming freely in water is the confusing part. The surface of a protein sees water, but a single molecule or just a few molecules, and then the next protein already comes. This is a better image. So it's just like clockwork, ingested or infused with a lot of water inside. So like high pressure water put into a solid state object. So in that sense, they're not quite right. Anyway, so back again to the background of the dimension. I said that already. The synchrotrons are piggybacking on the protein data now. This is here the Fermi lab, and this is just an amazing dimension. So you speed over many kilometers, or that is the one in CERN here where you have 27 kilometers, and this is in fact the circle in which you speed the X-ray beams. They go round and round and round. Okay, that's one of these tunnels, 27 kilometers of that. Just imagine this the size of that. Now we get into something completely different. Why not just look with a microscope at a protein? And ultimately the answer to that one is because they are too mobile. They change, or it's a scale that is just beyond the microscope, plus they are too mobile. What you get from that now is cryo, so you make them very cold, electron microscopy. So those are the scales of resolution that are accessible by EM. By cryo-EM. For a particular protein that is called GRO-EL, that's a protein that is forming a basket. So this is the basket, you see that, made of several units. Intego. Who's Intego backup assistant? That's Microsoft, isn't it? It's some strange program that interrupts. Yeah. So there was some program asking to be updated, a backup assistant or something, while you give a slideshow. Yeah. Anyway, so there are essentially two parts of a basket. Inside here is where proteins fold. That's GRO-EL. That's what GRO-EL does. That is on the level of 32 angstrom resolution, and some EM models in the PDB of those 430 are on that level, and some, the very, very, very best one today, do reach this level. What you see immediately from this level is that you still don't really see proteins on the atomic resolution. What you see is a surface. What you see is there are holes here. You see coarse-grained holes. You see more detail how you can reach into this basket. You see some aspects of the function clearly, but you don't really see the atomic resolution. In particular, you don't see inside. That's the limit of EM. And now, here, cryo-EM, what now happens is that you sort of have a combination of two worlds. In the combination of two worlds, you on the one hand have the EM images that are relatively low resolution, and on the other hand, you put in the comparative modeling solutions, the experimental structure that you have already. You do some modeling and try to fit these units such that they fit to that image. And that, in fact, is a task, is a world that maybe I don't give enough space in my lecture. This may be the most important aspect of structure prediction today. And in Munich, the group that is closest to doing something like that is the Max Planck Institute, Baumeister and these people. And they actually really push this theme and are a world leader in this theme. The modelers typically, so they use the modeling part for them comes from Modeler. I talked about it from Charlie. There's a collaboration between Baumeister and Charlie. But that, I believe, is a great place if this sort of thing would interest you to start. Okay, let's get back to an overview part of the talk. The Protein Structure Initiative. Structure genomics is the idea. And this is a befitting point in time because it is dying this month. Structure universe. So here the idea is we have the protein space, the ocean, and then we have solutions to the way protein structures form, the islands. There are two types of islands here, green ones and white ones. Green ones signify that I know experimentally that structure. White, I have no structure of this family, and then there are sort of half white, half green examples indicating that maybe I know structure here, but there is some additional helix or something like that, some additional motif, and for that part of the structure I don't know. Maybe they do something new in terms of function, that part I don't know. The goal, again, I talked about this, our understanding now is that this is more smeared out. Either way, the idea of structure genomics was, why not simply do a large-scale effort to go for all the white islands? To do a structure for every single protein family that has not been experimentally determined today. And ultimately, one of the background issues to that is, when I said that there are 100,000 proteins in the PDB, but when I reduce just at a level of 100%, almost 1/3 of them go away. So only 2/3 are such that they're not identical. And when I ask how many are such that they have less than 90%, there's less than half. So there's a lot of redundancy in it, because they grew in a way in which people care about a particular protein, and then they did another structure of that, another structure. That is because people care for that particular protein. Also because it's much easier to do a second structure than the first, because you already have the basic solution. And now all you need to do is sort of the fine-grained solution. So there was almost no example where a very high resolution structure, like 1 angstrom or 1.2 angstrom, was done as a first. It's always a follow-up solution. So a lot of that is like that. So this would then sort of change that and give a clear direction. It started in the US 15 years ago, where in fact at some point there were 4 large centers here, and they essentially had something like $250 million and produced about 3,000 to 4,000 structures, which meant that they created about 1 structure for $100,000, and that had been the goal with which they started. At the onset of structural genomics, the price had been much, much higher. Okay? That was one outcome of structural genomics. Structural genomics had many, many outcomes. And here's a bunch of these centers. Today, we have as many structures in 27 days as before in 27 years. And again, this statement is outdated. I don't know what really is today. I should rephrase. Most of those are not new, only a small fraction really is new. That is the sort of 8% number. Structural genomics received for 15 years about $150 million per year. And it ends this month. So the initiative is now finally over. There were 3 phases, and over the course of those 3 phases, this funding line was upheld. And our group was involved in the target selection. So again, the idea behind target selection, that is a slide from Christine Orengo, who was also involved in the target selection, was really that you have this major theme of a structure, and that major theme is repeated. So you have somehow like this, the circle here, the purple circle, and in another protein you have the purple circle, and then you have aggregations of different elements. And what we want to go for are these elements. Can we just do a 3D structure for each one of these elements? And what would be the problems that we would encounter? Well, the first problem is really to put the entire structure determination process into a pipeline. In fact, 15 years ago, nobody had ever done that. That was done. It was a real manual approach, and for every aspect of it, there was some expert who could do it, but nobody ever tried to sort of find a protocol that could be used by anybody. This then needed target selection, so which one should we go for? Cloning, so getting essentially from the genome sequence to the protein sequence. Expression purification, crystallization, diffraction, and structural analysis, essentially doing the structure. The last part I showed you, the beams here, a lot of that was computation, that is sort of this image, and some robotics in between. So again, the target selection begins by grouping. Essentially, in the space you have a bunch of sequences. Now, the first is that you have to say those are similar to each other, and by that similarity I can in fact say I already know the structure for that one. That's comparative modeling if you want. Now, I told you about the problem to define a metric in sequence space. And part of the metric issue is that I don't know distance. I can say protein A and B are similar. I cannot say that because A and B are not similar and A and C are not similar, I cannot make a statement about C and B. Because what my statements that come from alignment methods are essentially such that they only say similar when I see similarity. They don't say distance. And to have this space mapped out, I need distance. Okay? So I cannot do that. At best what I can do is say, okay, somewhere here my dark circle, my dark star, the star here is the structure that I know, for which I can do comparative modeling here, and I can find a circle around it, in which one, in that circle, here are 2, here's 1, for which I cannot do this, well, I could have a circle. But in a circle around it, I can do comparative modeling. And in this particular example here, those new sequences here, 1, 2, 3, 4, 5, I could not reach with today's models. Okay, The problem always is in overlaps. So whether the circle is like this, whether there's a larger circle, that depends a little bit on how I define my sequence similarity search. That could be the profile search, and what in yesterday's search came out as something that was not recognizable today may come out as something that is recognizable. And those are issues that we have with this. Some of the assumptions that we had at the beginning of PSI, so in 2000, 15 years ago. One is that, in fact, 2000 it started, we can do a structure for $100,000. I already showed you that we can. But that at that point was a assumption. They were all equally valid assumptions. We can automate structure determination. Crystallization is going to be the major bottleneck, because historically that's what the people had seen. The major problem that they had, for instance, to get important proteins such as membrane proteins, was that they couldn't crystallize them. And the idea was that we would have to go for about 1,000 to 10,000 structures. The next assumption was that prokaryotic biology is enough to get to all the biology in some sense, give or take a little bit, and that in fact X-ray suffices to get the main skeletons. And we could essentially do the whole thing in 10 plus years. Okay, I already told you it was 15 years, so 10 plus years by that criterion would fit in, right? It could still be true. This is true. We can do the structure for 100,000, and we can automate the structure determination protocol. And it worked very well, and in fact that is exactly what these pipelines did over 15 years. Now the first question, is crystallization a major bottleneck? And again, for many of these cases here, myoglobin is the answer. Now I know this is actually the Kendra structure here. For many of these, this was assumed to be the major bottleneck. Now, when I look at a larger set of proteins, so clone 6,000 in the PDB, so structures, that means 235. And what I'm looking at is how many did I get out into my pipeline, experimental pipeline. Of those, how many did express, how many were purified, and how many led to structures. And arguably, there is no one bottleneck here. At every single level, there is a major loss, right? And that was the first problem that people had absolutely not expected. That from clone to express to purified, that essentially every single step here would have such a big loss. Yes, crystallization is one major bottleneck, but not the one. It's not the only one. Cloning, expression, purification, they are equal bottlenecks. Let's get into the other issues here. And that brings us back to the target selection concepts. So again, we wanted to select these white islands. And initially, what we as the computational biologists were confronted with were experimental biologists who said, okay, so your task is very simple. All you tell us are families that are interesting to us and the ones that are going to work. Right? So tell us things that are new, and that if we in fact do put them into the pipeline, we will succeed. Can we do that? What would be your algorithm? What would be your concept? What would be your idea to deliver? It's a very simple order, right? It's understandable from their perspective. It sounds simple. You all understand it in a few seconds. Can we deliver? So we can close all the windows and then switch on the cooling. What will we do? So first of all, the first question is, how do I find out interesting families? Any idea? How would you find I give you all human proteins. How would you find things that are interesting here? Interesting and novel. Let's begin with the novel thing. So a novel thing is clear, right? Everything for which I can do comparative modeling, I throw away. So whatever is left is my search space. Now, how can I find things that are interesting in the whatever is left part? Oh.
Fire alarm's going off.
And that means?
I don't know. Fire?
Okay, so we have a lecture outside. This is a new complication. And for was to simply find out large families, build large psi-blast families here that are such that, in fact, this is not a good example. So empty families that are such that we have a huge family A, where we find nobody in the vicinity, but a huge set of things around it, and there's no functional annotation. Our argument was that if anything is reproduced in evolution so often that you have hundreds of sequences, it will one day turn out to be important for biology, even if it doesn't have an annotation today. And that, I believe, was absolutely true. But it's also true that it will take a while before the people, so most of these structures that were done on these, when they came out, nobody cared. 3 years later, people came back and said, yes, that's a cool structure. Now we discovered this is ubiquitin. And in fact, there are different ways in which proteins are digested. Structural genomics started an entire field of biology of digestion systems, simply because they did structures on things that are absolutely important. People had assumed that there's one way of digesting proteins. structural genomics uncovered there are many different ones, and they had not been labeled. So that is one example where they got a lot of credit. But most of these examples, they didn't get credit because somebody much later found out, and that wasn't too late to sort of couple directly to the grant success. That was the downside of that. Again, we can infer similarity, we cannot infer distance, we cannot find new folds. And now, let's just look at a few results of what we get. Another question here, by the way, was that we would only have to do prokaryotic biology, and the slides here are from Jinfeng Liu, and they're not quite new. This is residue coverage by comparative modeling in 2002 for a bunch of prokaryotes, archaebacteria, or they're all prokaryotes, bacteria, archaebacteria, and eukaryotes, entirely sequenced. So in the ballpark of the PSI-BLAST reach, you can model about 30% of the residues. Then part of the target selection task was to throw things out that did not work. And things that don't work, for instance, we know membrane proteins don't work, coil regions don't work, disordered regions don't work, things that are post-translationally modified, single peptides will not work. So all of this here we can exclude. This slider here goes again to something like in the ballpark of 20-something%. This is 30 plus 20. So we clearly know those things will not work. But for the remainder of the residues, for the remaining roughly 25 plus 30, the remaining 45%. Now the task still, the experimentalists still argued, give us the ones that succeed. And that clearly we could not deliver on. But again, in the overall numbers here, that's the disorder story, these two numbers add up. So 53% of the residues are to do, and those we do by simply this target clustering that I showed you, essentially indicated to you before. And there are some issues with domains, so we need to base this on domains, and that in fact is a big issue with the clustering, in which we had to do it. At the end of the story, for the cutting of the domains we talked about, right? That was something that we had to develop, but it was not easy. Now, again, let's look at how structural genomics is changing the coverage of the universe. Essentially, I'm trying to answer the question, how many white ones did we add? And it turns out that this is a very difficult question to answer. It sounds so easy, but it's not. So the first question here is, how do I compile coverage of wide domain, so to speak? So one way could be I look at the entire protein. Another way would be I look at fragments. So I cut it down into fragments, and then I say win or coverage is 100% if each fragment is covered. And another one is I say that all the residues are covered. So in the first one, protein coverage, I mean that any region in the protein for which I have a structure, I say this is covered. Okay, tiny bit is covered, I say protein done. The green one is the most extreme, where I say every single residue that is not covered is not counted, so I need to really get the entire thing. And here I say that if I only had this one, I would say 50% of the fragments, only this one 50%, both of them 100% of the fragments, even if these regions are not done. But I can cut it into two major regions, I get two major structures, end. Okay, so that's the difference in the numbers that I'm going to show you. What I show here is number of domains, and I sort by the largest family. And I'm asking what fraction of coverage is taken, and what I'm looking at here is the differential to when we started. So everything for which we could not do comparative modeling. And I asked from today to 15 years ago, how many do I have to do? 10,000, 20,000, blah, 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 you see? And the first sort of 2,000 or something like that gave, green is per residue coverage, proteins covered, fragments covered. So 40% roughly is the first 3,000. And you see how it continues. So the idea at the onset that we would get away with doing 1,000 structures and then cover 100%, that is already clear at the point at which we got to this clustering here was absolutely not true. There is one issue here, and that in some sense leads back to what you said. about the first, at that point, 40,000 or 38,900 were clusters, were groups of 2. Everything there was singletons. So there were these cases where you have, in one organism, you find the sequence. In fact, if you look at all the clusters, if you looked at all the families, most of those are singletons. It's just a number game. Ultimately, we believe that singletons will become families if we just sequence enough. But when you, for instance, the first genome was sequenced 1995, so 20 years ago. And there was a certain fraction of proteins in that genome for which we had no similar protein out there. Okay? Now we do a lot of sequencing. And many of these that we previously did not find, they were called orphans. They were previously called orphans. Today should be no longer orphans, right? And that is true. But now, what do you believe if I did plot the fraction of orphan for every genome that is added over the last 20 years? Is that changing? So we sequence so much today. Do we get fewer and fewer orphans? So again, if I looked at the Haemophilus influenza orphans from 1995, most of those, I would guess, if not all, I really don't know the number, most of those will not be orphans. I know it for human. So the 2000 human version had 10% orphans, and today, of these 10%, that would correspond to something like 2,000. And today, the number of human families that are still orphans is more in the ballpark of 100 something, 200. So strictly much less. But now I'm asking something else. I'm saying I'm digging out new organisms to sequence. And for every new one, I'm not asking for the ones that I did 20 years or 10 years or 15 years ago. How is the orphan numbers changing? Now I'm asking for the new ones that I take out, that are not really related to things. Okay, if I do human again, then that is clear. But most of the things that are done today, in fact, are fairly different from, or many things that are done are fairly different from things that we had before. So what is the orphan ratio? Is that going to 0? Is my question to you. It's surprisingly constant. So completely surprising statement. That in fact is what this kind of thing would suggest, that you get to a point where you converge, that your orphan ratio, at some point we would reach 100, maybe a long way away, but at some point we would cover it all. But looking at the sequence studies of new sequences that are added, it seems that every soil bacterium that I add adds so much novelty that again, if I do another 10 years, I will bring in all its family members. But today is still so much novelty added that we are nowhere near saturation. In fact, the ratio is today not much lower for the new things than it was 10 years ago. That's totally surprising. And that invalidates the statement. So this statement is still operated under the assumption that we somehow converge, and that the set that we had then, this is based here on about 1 million sequences, that this set would be relatively complete. Okay, this is an invalid statement. But even in this invalid statement, you absolutely clearly see there is no cheap way out. We absolutely need many here. 10,000, so we thought 1,000 would be enough, but even if we did 10 times more, then they would only cover half. Well, now you could say half of what is left is a big step, if we could do 10,000. And that clearly is true. The top 2,000 would cover already 1/4 of that. Now, well, most proteins are multi-domains, and that actually was found out in context of this project. Now, let me just fast forward to, no, maybe it comes later. Here's another statement. Everybody had assumed that when you get a structure, sequence determines structure, determines function. So when you get a structure, that will teach you what the function of the molecule is. And this is the answer of Claudia Bertanato and Sharon Goldsmith-Fishman in the Barry Honig lab, in which they essentially, what they did is, Sharon and Claudia, they looked at every single structure that was determined and spent many years, these two, on trying to assign function. reading all the literature that they could, study whatever they could study, use whatever tool, and then call up once they had a hit, meaning that they had a suspicion about what this thing does, that was previously unknown, that in fact, then they called up the experimentalists and said, listen, we have a structure. And struck up a collaboration. Now, the surprising reality is that most of those for which really function was not known, so we had a large family that was functionally not annotated, and structure did not provide an annotation. It's as simply as that. So most of those, from 88, 60 remained unannotated. Only for this fraction, the structure clearly held something. Of all the novel hypothetical, there was some sort of idea that we could annotate something, but none of that was really clear. Okay, the green part is, you could have not done without the structure, so you clearly can do something you couldn't do, but it's a speculation. Okay, we learned something, but not as much as we thought, and we clearly had assumed that the blue would be higher here. So that a lot of the hypothetical proteins would become named entities. And all that we could do, that they could do, and they really did a very good job, all that could be done was really to give an idea what people should try in the experiment. But then you have to find the right person, because it's an idea. If you have a name, you know who to contact. If you have an idea how to do an experiment, then that's more complicated. This one here is talking about novel leverage. What I'm talking about is now I deposit a structure. And I ask, with that structure, how much comparative modeling could I do that I could not do before? Okay? So in this is the leverage for the entire PDB. This is the leverage for a particular protein. And in this particular protein here, if you add that to the structure, this little green part and that protein here, that little green part, are the things that are new. This I could have done before, that part of the protein I could have done before, because it's similar to another region in the protein, maybe a similar domain, maybe a similar region, that may just be the helix, that may be a helix turn helix, something very key motif that is new. Okay? So now I'm going to ask, over the years, how much novel leverage has been done by the structures that were added to the PDB? That's shown here. And novel residue, novel proteins, PDB chains. That is just the number of proteins deposited in the PDB grows. This was an incomplete set, that's why this number is not higher. In this one here is number of novel proteins, number of novel residues. It turns out that in 2005 already, in the first round of structural genomics, every fourth of the novel structural deposition was done by structural genomics. And that was a lot. So I talked about $250 million. This is a lot, but compared to what the entire structural biology consumes in the US, this is tiny. This is more in the ballpark of 2% than in the ballpark of the 25% this one indicates. So this was an amazing bang for the buck in some sense. And that was clearly a success. How many clusters? Clearly, so the idea of 1,000 was not enough. What about 3,000 to 16,000? That is what I showed you before. Maybe that would give us sort of, remember I said that this would give us a fourth, this would give us a little bit more than half. But the question remains, would prokaryotes suffice? It used to be the answer was yes. So now what I'm going to say, what I'm going to do, is I'm going to slice. So again, this is sorted by the number of families, the size of the families. And I'm going to say, for the first 500, for the largest 500, what fraction of those is prokaryotes and what fractions of those is eukaryotes? Okay? And then I'm going to, essentially, for each slice, going to give you an answer now. And that will, in fact, show that we need more and that prokaryotes alone is not enough. Here are the slices. The slices are families larger than 1,000, 2, 3, 4, 5, maybe some counting mistake. 2,500, 1,250. So in all the families that are that have 1,250 members and more, no, in the in the 1,250 largest families, about half of them, or 1/3 of them, are mixed, meaning they have prokaryotes and eukaryotes. 1/3 is only prokaryote, and 1/3 is only eukaryote. you see that the mixed families, in fact, go flat very soon, while the eukaryote-only families shoot up. Why is that? Do you believe? So again, what I'm showing you is something that has never been published, something that nobody in the world expected. We just never had the time to write this up. And now I'm asking for an explanation for something that is surprising to everybody. This is how I am, right? Why?
I would say eukaryotes are more complex and have more kind of complex cells and maybe transmembrane
Yes.
protein stuff. And
But it's not true here. So, I'm going to say something that is not quite true. By now you know that I enjoy getting you in the wrong direction, right? But the smaller families are the singletons more. And so now what I'm pushing is this image that eukaryotes have a lot of new inventions. They may increase their complexity. Your argument may still be right. Is that the reason, is now my question, of what I see? In some sense, clearly is. But what I'm fishing for really is the simple statement is that the largest, it's sort of trivial, the largest families are such that they are across all kingdoms. That makes them large, that they share all the kingdoms, right? And they are ubiquitous, they are everywhere. That's why they are so large. Okay? And of these, in fact, we have a lot of structures, or we cannot do them. Membrane proteins, you mentioned one of those. So that means they are left out here. Because what I'm looking at is whatever remains today in 2005. And from there on, there were some large families left, but not many. So the moment we don't have mixed, it's clear that we go either red or blue. That part is already trivial. That the moment this one goes off, it has to go into the separate paths. Why now the eukaryote is higher than the prokaryote? This is unclear. But the main finding here, or the main clear reality is, doing prokaryote structures is not enough. Okay? Why would we want to do prokaryote structures? Why was there ever this argument that we want to do with prokaryotic biology all the eukaryotes? By the way, this is a cutout. Here is the full plot. Both together. Why do we want to do prokaryotic biology for explaining eukaryotes? It's easier. So putting a eukaryotic protein into the tube is much more difficult. Every aspect of it. The cloning, it begins with the cloning, but I said for structural genomics what you need is cloning, expression, purification, every step for eukaryotic protein. So NASG, the group headed by Guy Monteleone, did the effort. They looked at 100 human proteins and they compared them to the prokaryotic homologue. And for on average, the effort they needed to get to the same result was 10 times more than 10 times more for every single one. So rather than 100,000 for a structure, that means you pay $1 million for a structure because it's a eukaryote. Okay, and that is of course a challenge. So if you can do that with prokaryotes, you want to. And here's the limit of that. We have reached the moment when prokaryotic biology no longer is enough. But anyway, so we need more fragments. Here are some other problems that I don't want to get into. But largely, I believe structural genomics was clearly a success. Here is another part of the Edda Kloppen, some of you may have seen her, went on into a consortium that did the same for membrane proteins, and I will talk about membrane proteins at some other point. I go over this. And there were some fantastic structures done by this membrane structure consortium, and some of those, it's not really, this is an example where we select one from the Wayne Hendrickson group. This is one of those beautiful stories here. It was published 5 years ago in Nature. For this particular structure, that was the highest resolved membrane protein ever. It was a new structure. So I said, usually when you have a high resolution structure, you don't have a new one. This was a new one. It's 1.2 angstrom for a membrane structure. Incredible. So you do the phenylalanine ring. You saw everything in that structure. Something totally new, and on top, they had all the biology in it. So the SLAC1 was an example where the prokaryotic structure told you something about the way light is taken on by a plant. Something that a prokaryote absolutely doesn't have. But the biology was explained by that structure. It's just one of those moments in history that I found remarkable. That was just one of those success of this NICOMS consortium. That's the overall selection, target selection, and cloned, expressed, purified from different groups here. And that's the one headed by Wayne Hendrickson, the blue. The red is headed by Wayne Hendrickson and 8 others, and you somehow see how the blue is impressively dominating. But anyway, so let's get through that. Most membrane proteins we still don't in fact have a structure, and that is one of the challenges that remains even after 10 years of doing this. Okay, that's thanks to Edda and Marco. Let me get one back. That's Wayne Hendrickson. Those are people who were involved in the pipeline. Brian Kloss, James Love, Renato. They did the pipeline at the New York Structural Biology Center that I showed you before, the NMR machines. Larry Shapiro, Ming Cho, Filippo Bancha, they did structures. Let's get back to definition. 5 minutes. Tour de force. Otherwise I never get to my secondary structure and stuff. Just a quick one. You get 3D coordinates. From 3D coordinates you get to secondary structure, and the background idea is from Linus Pauling, hydrogen bond stabilized secondary structure, and essentially he is one of those people who got 2 Nobel Prizes, one for the hydrogen bonds story and one for the peace, for something very different, so one in chemistry, one in peace. Here some of the publications. I have briefly mentioned that. So there is a PNAS issue in 51 and another PNAS issue in 53. In these 2, he published what I show you is 1, 2, 3, 4, 5 papers. There are way more. So he published 15 papers in these 2 issues. And each one of these papers describes one theoretically possible result of a hydrogen bond formation. So one type of secondary structure that is formed using that hydrogen bond. 15 papers, 13 were wrong. That was before the structures were observed. Mind you, the first structure was done 59-60. Most of those 15 were never observed. 2 were. That is a Nobel Prize. So that's all you need to do, right? 2 in 15 right in a crucial topic, and you get the Nobel Prize. So John Kendrew, Max Perutz did the first and second, officially celebrated as simultaneously, although not quite simultaneous as you see in the dates, myoglobin, hemoglobin, both in Cambridge, which is remarkable, right? That the whole world is trying to get crystal structures. 2 guys in Cambridge get it on myoglobin and hemoglobin, and there was no reason. And they are very, very similar. There are reasons, and it's not a mere coincidence. But they actually did not take it because the other one was working on a similar protein. There's just the way they got them into high concentration was essentially the reason why they chose those. Now, looking at these, you understand what the people believed in the 60s about secondary structure, right? All helical. So after that, people believed Linus Pauling was really completely wrong. He was only right in one of his, and all that you ever observe are alpha helices. Anyway, how do we assign secondary structure from 3D coordinates? And one solution uses essentially the same idea. You use hydrogen bond formation. One, in fact, looks at geometry. So you have long cylinders, you have these sheets forming, and that's geometry. That is defined. The model, the idea of DSSP really is to use the hydrogen bonds from Linus Pauling. The Dictionary of Secondary Structure of Proteins, the DSSP stands for, and so the major two people here is Chrysander, a giant in the field. So this is for you. So Chrysander. Over 6,000, one paper, the DSP paper of 6,000 citations, H index of a 92 ICB, International Society for Computational Biology fellow, just an immensely important guy with a lot of vision. The other person on it is Wolfgang Kapsch, that's the paper, and that still, 83, is still the method that most people use to assign secondary structure. And the way it works is simply you use the Coulomb energy model that defines, the Coulomb energy defines a cutoff between two proteins, that defines the hydrogen bond. Once you have the hydrogen bond is defined by some threshold in the energy. If the energy is higher or lower, they call it a bond. And then you have a motif of bonds. I and I plus 4 is a helix, or I plus 3 is a 310 helix. So the beta strands are defined equally at adjacent strands. And this way you essentially do it. There are other methods. One comes from Dimitri Fischmann. I'm not going to go into it from Wein-Stefan, from Freising. I'm not going to go to it, because ultimately what people use today is DSSP. One issue with secondary structure is that here's shown calmodulin. Upon ligand binding, it changes its secondary structure. And another issue is, a related issue, is that there are regions here, shown in an NMR structure, where we have motion, and in this motion, secondary structure may not be well defined, other regions that may be defined, so secondary structure may differ according to how strongly defined a certain region is according to dynamics, according to the function of a protein. And those dynamics are measured, that is done by Arthur Palmer here, wrong quote, this is from the paper, but he invented this autoparameter. And this autoparameter measures the degree to which you have a wobble, the degree to which you observe motion in a protein, in a native protein. Arthur Palmer is an NMR spectroscopist. So what I'm going to show here is the NMR model variation versus the degree of motion. That is this autoparameter. And you see that if you see more variation in NMR models, you see higher degree of motion. Meaning that when you observe in an NMR model a lot of wobble, that translates to biological movement. Because this is the right way of putting it. So if you had points here, then that would be the example of the NMR model is wrong. They just can't define their right model. But essentially you see that there is a clear relation. Now, the question is, can we see that on the level of secondary structure? How would we see that? Well, you could change the way DSSP is computed. But we took a different path. We simply said, well, the problem really is in some sense that there is, secondary structure is the idea of having one thing, one state. And that is a mistake, because if you wobble around, you may not have that state. So rather than changing the way DSSP is done, change the way we look at it. So rather than thinking about secondary structure as something that has a letter, think about it as a letter with a probability. Each state has a probability. So in that context here, this particular position would have a certain probability of being in the S on this strand. So S for in DSSP is non-regular. E is extended as a strand, red is the helix, so it has a certain probability of different positions of this strand have a certain probability of being in different states. And the probability at each position of the residue is measured by this letter. So this is only loop, this is only strand, this can be strand or other, this can be strand or other, but this is more strand than this, and so forth and so on. So every single position now has a probability vector for secondary structure. So continuous assignment, and the way you do that is you simply use the same Coulomb formula, but the Coulomb formula has a cutoff, and the cutoff essentially assumes that water has a certain radius, a water molecule. And now we vary a little bit this water molecule radius, and the work is done by Klaus Andersen, who is now at Siena since many years, Siena Tech in the company. And so you simply take DSSP, the hydrogen bond, then you put a weight for different sized hydrogen bond thresholds, and you have some. So we essentially, rather than having one threshold, we have 10, and we weigh 10 different DSSP runs at a certain way, okay? With 10 different thresholds. And that gives us a probability. At every single position, you get a probability for one of the 7 DSSP states. And now the question is whether that probability, that's the way to choose the vectors. Now, what this one says, let's look at this one here. The degree of motion, and here is sort of the degree to which secondary structure has one assignment. So down here, we see that secondary structure is clearly defined. Up here, you see secondary structure, no, it's the other way around, wait. No, up here is clearly defined, down here is not clearly defined. So when you have a residue that can be in different secondary structure states, that's down here, that's where you see motion. When you have a residue that can be only one secondary structure state, that's when you see no motion. This parameter here is done by an experiment, is done experimentally. This is just looked up from a model. Now the remarkable thing here is, now what we can do is we can assign continuous secondary structure for a crystal structure. Okay? And we can now look at a crystal structure and an NMR of the same protein. And NMR gives us different models. We can call that a continuous secondary structure. The crystal structure gives us only one model, and we can compute continuous secondary structure by that. And then we can compare. And it's remarkable how similar these are. What that tells you is that in fact this idea that NMR tells you about mobility is true. But when the NMR structure is done well, it is absolutely true, but actually it's also in the crystal. The information about flexibility is in a crystal. All you need to do, so I'm not even looking, I don't know whether you have ever heard of R values. So they somehow measure B values, R values. B value is a local flexibility measure that X-ray crystallographers provide in their structures. I'm not looking at that. All I'm looking at is what's the probability of the, what is the move space for every single residue and the position in which it is in that crystal structure. And that tells me something about flexibility. You can directly translate that into something that somehow correlates. And that's remarkable. And I found that was remarkable in the context of secondary structure assignment. For the time being, this is where I stop. Actually, that is where the lecture stops anyway. And from here on, we will go, I will skip that part, and we will go directly into secondary structure prediction methods. Thanks for your attention.